Good afternoon to all the EAPCET aspirants. So here we are with the major chapter, that is respiration in higher plants. Okay. So this chapter includes the concepts which are here, where we need to deal with the major concepts from this chapter, which are very important for your EAPCET as well as for your NEET as well. So in the beginning of this chapter, you have to know the clear differentiation between breathing and respiration. Second is, what are the respiratory substrates? You have to know the clear idea on what are the respiratory substrates. Followed by cellular respiration, types of respiration based on the respiratory substrate, types of respiration based on respiratory substrate, okay, and based on respiratory substrate and based on the presence or absence of oxygen. And next we will look into the glycolysis, which is a 10-step process operated both in prokaryotes as well as in eukaryotes. And then is anaerobic respiration. Here we will be exercising both alcoholic fermentation and lactic acid fermentation. And then in aerobic respiration, the immediate step after glycolysis, which is operated in mitochondrial matrix, will be oxidative decarboxylation and Krebs cycle. And the last step of aerobic respiration will be electron transport system. And then at last you will look into amphibolic pathway and respiratory quotient. So let's begin with what is the difference between breathing and respiration. Breathing is the general exchange of gases, right? Breathing is the exchange of gases, whereas respiration is oxidation of macromolecules and liberating out the energy. Now you got the clear difference. What is respiration? Respiration is nothing but oxidation of biomolecules, or you can say biomacromolecules. Biomacromolecules. These biomacromolecules can be proteins, carbohydrates and lipids. Fine, right? Those macromolecules which are subjected for respiration and they liberate energy, these macromolecules are also known as respiratory substrates. Respiratory substrates. Fine, right? So these are the respiratory substrates, and you got the clear definition on what is respiration, right ma? And now we will look into cellular respiration, the concept of cellular respiration. The definition is as simple as it is: oxidation of macromolecules inside a cell and liberation of cellular energy after oxidation is known as cellular respiration. Oxidation of macromolecules inside a cell and liberating cellular energy. Cellular energy is ATP. Okay, the energy which is released after the oxidation of macromolecules is stored in the chemical bonds, that is ATP, which is the direct energy used for the metabolic processes or the metabolic activities that are happening inside the cell. This is cellular respiration. Now I hope you are clear on the terms of respiration, respiratory substrates and cellular respiration, right? And now we will look into, do plants breathe? Of course, we have learned the difference between breathing and respiration. Now we will see, do plants breathe? Breathing means what? Exchange of gases. Of course, a complex respiratory system and the breathing mechanism is present in all the living organisms, be it through gills or lungs, fine, right? But in plants, if you see, there is no complex respiratory mechanism, but then for the exchange of the respiratory gases, it possesses stomata and lenticels. Lenticels on stem, over stem, and stomata over leaf. Fine, right? They will promote the exchange of gases. So here if we see, they will promote exchange of gases. Why there is no complex respiratory mechanism inside the plants? Because there are three prominent reasons. One, the entire plant body is composed abundantly with one simple permanent tissue, that is parenchyma. These parenchymatous cells are loosely arranged inside the plant body. They are loosely arranged. They will be possessing the intercellular spaces where the gaseous exchange or diffusion of gases will be simple. And every plant part will take care of its own gaseous exchange. Second reason is every plant part takes care of its own gaseous exchange. Fine. And third important reason is all the cells which are live and active, okay, live and active cells, most of the cells are closely appressed to the epidermis, they are close to the atmosphere. Fine, right? The internal tissues will be mechanical tissues which will be dead, and their main purpose is conduction of water and minerals. Okay, apart from that, all the cells which are involved in the photosynthetic activities, they are close to the atmosphere. Fine, right? And internally located cells will be mechanical tissues. Most of them are dead mechanical tissues, and they will be conducting water and mineral nutrients from top to bottom of the plant. That is the third most reason. These are the three major reasons that plant system is lacking the complex respiratory organs. Fine, right? It has just stomata and lenticels for the gaseous exchange. Clear till here? Any doubt anywhere, ma? Now, I will give you a clear framework on the respiration, both plants and animals, and then on the bacteria. What is the difference between anaerobic, aerobic respiration, and then I will clearly make you out on the differences of the respiratory substrates. Here we look into the types of respiration. Okay. Very important, ma. Types of respiration. Fine, right? We have different types of respiration. First, types of respiration based on the respiratory substrates. Based on the respiratory substrates. We have two types of respiration. First will be protoplasmic respiration and second is floating respiration. Fine, right? Protoplasmic respiration and second will be floating respiration. In protoplasmic respiration, what will be the respiratory substrates? Respiratory substrates will be proteins over here. Here proteins are the respiratory substrates. And in floating respiration, if you observe, what will be the respiratory substrates? Fats or lipids and carbohydrates are the respiratory substrates in the floating respiration. Fine, clear till here? And then you see second is based on the presence or absence of oxygen. We have two types of respiration here. First will be anaerobic respiration and second is aerobic respiration. And then aerobic respiration. Anaerobic respiration is mostly operated in prokaryotes and aerobic respiration is mostly operated in eukaryotic cell. So here we see what is the common pathway for both aerobic and anaerobic respiration. The common pathway for both aerobic and anaerobic respiration is glycolysis. Glycolysis is the common pathway for both aerobic and anaerobic respiration. Fine, right? The end product of glycolysis will be pyruvic acid, and here the pyruvic acid is subjected to, or pyruvate is subjected either through lactic acid fermentation or alcoholic fermentation. Lactic acid fermentation or alcoholic fermentation. Fine, right? And then is aerobic respiration. Aerobic respiration, here we go with the process. So here you see, in aerobic respiration what happens if you consider this as a cell completely. Okay, consider this as a eukaryotic cell, and this is the cytoplasmic compartment followed by mitochondria. Fine, right, ma. So here inside the cytoplasm, glycolysis will happen, and this will be releasing out what? Pyruvic acid. Pyruvic acid after glycolysis will directly land into the mitochondrial matrix. Here let me give you a clear view on 4 components of mitochondria. Here if you see, this is the outer mitochondrial membrane, and this will be inner mitochondrial membrane or cristae. Fine, right? And this will be intermembrane space. Fine, right? And inside which you will be having mitochondrial matrix. Fine, right? So here pyruvic acid, when it enters into the mitochondrial matrix, the immediate steps which happen is, first and foremost step that is happening is oxidative decarboxylation. Oxidative decarboxylation, wherein 3-carbon pyruvic acid is turned to 2-carbon acetyl CoA. This acetyl CoA is then passed on to TCA cycle, which is tricarboxylic acid cycle, which is also known as Krebs cycle. Post which in inner mitochondrial membrane or cristae, third step or the last step of aerobic respiration will happen, that is ETS cycle, electron transport system, where the complete energy, the complex energy, be it NADH, FADH2, which is formed at the end of Krebs cycle, will be turned to the simplest form of energy, that is ATP, which is the cellular energy. Clear till here, ma? Any doubt anywhere? Fine, right? I hope you got a clear framework on types of respiration. Once again, I will repeat to you. Types of respiration based on the respiratory substrates. If the respiratory substrate is proteins, we will call it as protoplasmic respiration. If the respiratory substrate is fat and carbohydrates, it is floating respiration. and then based on the presence or absence of oxygen, that is oxidation of macromolecules in the presence or absence of oxygen, based on that we have anaerobic respiration and aerobic respiration. Of course, the anaerobic respiration is completely operated in a single compartment, that is cytoplasm. Fine, right? So inside cytoplasm only glycolysis will happen, alcoholic fermentation or lactic acid fermentation based on the cells, based on the prokaryotic organisms, or in eukaryotes also lactic acid fermentation happens inside the muscle cells for the immediate requirement of energy. Fine, right? So this happens inside the cytoplasm itself. Whereas aerobic respiration, the sequential process of oxidation and sequential release of the energy will occur in aerobic respiration, and complete process will not happen inside a single compartment. Okay, complete process of aerobic respiration will not happen in a single compartment, that is cytoplasm. It happens inside the cytoplasm, and then glycolysis occurs inside the cytoplasm, and then further steps are being done inside the mitochondria. Look into the components of mitochondria. You have outer mitochondrial membrane, inner mitochondrial membrane is also addressed as cristae, and then you have mitochondrial matrix and intermembrane space. Inside the mitochondrial matrix, if you see, two steps will be followed up after glycolysis. First will be oxidative decarboxylation, which turns the 3-carbon pyruvic acid into 2-carbon acetyl CoA, followed by TCA cycle or tricarboxylic acid cycle or Krebs cycle, and then the last step will be electron transport system. Clear till here? Any doubt anywhere? And then, yeah, here first we look into the glycolysis process. This process is clear to you? Yeah. Now we'll look into the glycolysis. Glycolysis is also addressed as, glycolysis is operated in the cytoplasm, which is the common pathway both for aerobic and anaerobic respiration. This is also addressed as EMP pathway. EMP pathway. Okay. After this is named after 3 scientists, that is Emden, Meyerhoff and Parnas. These 3 scientists, after these 3 scientists, it is named based on their discovery. Okay, glycolysis is also addressed as EMP pathway. And where does it occur? Both in a prokaryotic or eukaryotic cells. The site of occurrence of glycolysis is cytoplasm. Okay. Glycolysis will be 10-step process. The first 5 steps are known as energy requiring phase, and next 5 steps are addressed as payoff phase or energy releasing phase. So what are those 10 steps? We will look into that clearly. In the first step, glucose is phosphorylated. First step is phosphorylation. Phosphorylation. And this is by the enzyme hexokinase. Excuse. So the first enzyme is what? Hexokinase. Fine, right? Glucose immediately reaching up to the cytoplasm, it gets firstly phosphorylated at the 6th carbon into glucose 6-phosphate by the enzyme hexokinase. Here the phosphate group is added to glucose from ATP. ATP is required for the first step, and the second step is the isomerization reaction. Okay, second step is isomerization reaction. Isomerization. Okay, by phosphohexoisomerase is the enzyme here, where glucose 6-phosphate is isomerized to fructose 6-phosphate, and after which fructose 6-phosphate is again phosphorylated. Third step is again phosphorylation, where fructose 6-phosphate is phosphorylated at the 1st carbon and 6th carbon, which is turned to fructose 1,6 bisphosphate. To add a phosphate group, again at the third step ATP enzyme is required, and the most prominent enzyme of glycolysis here at the third step is phosphofructokinase. Okay, most prominent enzyme over here is phosphofructokinase. Fine, right, ma? So here the enzyme phosphofructokinase is known as pacemaker enzyme. Phosphofructokinase is addressed as pacemaker enzyme. Fine, right? Why is it known as pacemaker enzyme? And this step is addressed as rate limiting step. Very important, ma. You may get statement based questions. Why phosphofructokinase is addressed as pacemaker enzyme, and why is this step known as rate limiting step? It is because the third step of glycolysis, which is catalyzed by the enzyme phosphofructokinase, that is from fructose 6-phosphate to fructose 1,6 bisphosphate, will decide the accuracy or the speed of glycolysis. For example, in any eukaryotic cell inside human body, let us consider, during fasting days glycolysis will not happen, gluconeogenesis will happen. Fine, right? From the stored food glycogen, glucose will be formed during fasting days to survive or sustain on that day. When you are having high meal dates, okay, high meal is being consumed on those days, blood glucose levels will be rising up, shooting up, and glycolysis will happen to the maximum. So based on the requirement of the cell, glycolysis speed or accuracy is being monitored by what enzyme here? Phosphofructokinase, which is the pacemaker enzyme, and that is why this step is addressed as rate limiting step. Clear? Understood till here? And then fourth step is cleavage. Fourth step is cleavage. It is done by the enzyme aldolase, where fructose 1,6 bisphosphate is cleaved to 3 carbon compound, that is glyceraldehyde 3-phosphate and DHAP, dihydroxyacetone phosphate. Fine, right? Both are 3 carbon compounds only. Fine, right? And this 3 carbon compound from G3P and DHAP, this can be isomerized. DHAP will further get converted to G3P and continue with the reaction. So from the fifth step, that is from sixth step onwards, we have to calculate for 2G3P. For 1 glucose, here we have to calculate for 2G3P, because DHAP will be isomerized to G3P and further this will continue the reaction. So from here our countdown should be 2G3P. Understood, everyone? And here the fifth step is what? Again, isomerization. Now G3P is converted to 1,3-bisphosphoglyceric acid, that is sixth step, which is oxidation. And this requires NAD plus, which is reduced to NADH plus H plus. This you can say redox reaction also, because energy equivalent, that is NAD plus, is reduced to NADH, whereas substrate G3P is oxidized to 1,3-bisphosphoglyceric acid. Fine, right, ma? And now seventh step, that is 1,3-bisphosphoglyceric acid is turned to 3-phosphoglyceric acid, which is dephosphorylation. And here ATP is being formed. Phosphate group from substrate is given to ATP. That is the reason seventh step is also known as SLP, that is substrate level phosphorylation. Clear till seventh step? Eighth step you see, where 3-phosphoglyceric acid is turned to 2-phosphoglyceric acid by the enzyme phosphoglycerate mutase enzyme. And this step is isomerization. Fine, right? And then ninth step is dehydrolysis, where water is being lost at this step. Fine, right? By the enzyme enolase. And 10th step, where phosphoenol pyruvate is converted to pyruvic acid, which is the last product of glycolysis. Fine, right? Here also one ATP is being utilized from the substrate, one phosphate group is being taken from the substrate, and ATP formation occurs here at the 10th step. At the 10th step, ATP is being formed. Fine, right? So this is also substrate level phosphorylation. 10th step of glycolysis is also SLP, substrate level phosphorylation, because phosphate group is taken from the substrate and it is given for the formation of ATP. Clear till here? Any doubts anywhere, ma? Glycolysis completely it is clear, no? Now we need to draw out the bioenergetics of glycolysis. Look at here. Bioenergetics of glycolysis. First you have to know what are the three irreversible steps of glycolysis. First step, third step and 10th step. These three are the irreversible steps of glycolysis. Okay, what all we are discussing now, they are very important for your statement based questions, ma. Take down it clearly. Okay, three irreversible steps of glycolysis. Three irreversible steps of glycolysis will be first, third and 10th steps. Fine, right? And what is the rate limiting step? Third step is the rate limiting step. What is the phase maker enzyme? Phosphofructokinase is the phase maker enzyme which is catalyzing the third step of the reaction. So here we see the bioenergetics of glycolysis. Fine. First, look at here. First and third steps. Here energy is being released. 2 ATP is released into the cytoplasm. Fine. At 6th step, NADH is being released into the cytoplasm. Fine, right? First and third steps. 2 ATP are being utilized, because they are from energy requiring phase. 2 ATP are utilized from cytoplasm. And 6th step, NADH is released. For 1 glucose we are calculating 1 glycolysis step. So here 2 NADH are released because we are doing it for 2G3P, 2-glyceraldehyde 3-phosphate, right? DHAP will be turning to G3P again. So we are counting for 2G3P. So here at 6th step, 2 NADH are released into the cytoplasm. Fine. And this step is the oxidation process. And at 7th and 10th steps, 2 ATP each are released into the cytoplasm. Fine, right? On total it is 4 ATP. These steps are what? Substrate level phosphorylation steps. Very important. 7th and 10th steps are SLPs, substrate level phosphorylation steps. Fine, right, ma. So this 6th step is NADH, 7th and 10th step 4 ATP will be released out. Now here you have to know what is the total ATP, total energy released during the glycolysis, what is the net ATP and what will be the gross ATP for single glucose molecule entering the glycolysis process. Okay, what is the net, gross and total ATP of glycolysis is important for you. Look at here carefully. Yeah, first, net ATP of glycolysis will be 2 ATP, because from 4 ATP which are released into the cytoplasm, 2 will be utilized by energy requiring phase, right? So net ATP will be 2 ATP. Next is gross ATP. How much is gross ATP? Gross ATP, if you see, total amount of ATP that is released into the cytoplasm. Total amount of ATP is 4 ATP that is released into the cytoplasm. Fine. Next is total ATP. Four ATP which is released along with NADH. One NADH is equal to 3 ATP in our calculation, right? Approximately 1 NADH will be equal to 3 ATP. As such, we have 2 NADH released. So 2 into 3 it is 6 ATP plus 2 net ATP. So total ATP release is 8 ATP. Clear? This will be the complete bioenergetics of the glycolysis. I hope all the steps, processes, bioenergetics, irreversible steps, substrate level phosphorylation, everything is clear with respect to glycolysis. If you are having any doubt, you can please drop a message, I will address it. Fine? Clear till here, ma? Now, pyruvic acid is formed. We will look into the anaerobic respiration first. Okay. Yeah. Pyruvic acid which is formed inside the cytoplasm. For the anaerobic respiration, it is either undergoing lactic acid fermentation or alcoholic fermentation. First, you have to know in what type of cells lactic acid fermentation will happen and in what type of cells alcoholic fermentation will happen. In the unicellular organisms, like you consider yeast, which is known as brewer's yeast or baker's yeast, where carbon dioxide is released at the end of the alcoholic fermentation. Okay, this is used in baking and brewing fermentation processes. Okay, in such type of cells alcoholic fermentation is being used, and lactic acid fermentation is done in the most noted bacteria which is helpful for conversion of milk to curd, that is in lactic acid bacteria, and in the muscle cells of the living organisms for the immediate requirement of the energy. For the immediate requirement of energy, whenever during vigorous exercise times in muscle cells, when vigorous amount of energy is required, high amount of energy is required, immediately it is supplemented by lactic acid fermentation, and people will get muscle cramps after the vigorous exercise. It is because of lactic acid accumulation inside the muscle cells. Clear, right? And it happens in the muscle cells, usually inside eukaryotes. Now we are clear on where does this occur, right? Now we will get into the process in detail. So immediately after pyruvate which is formed, pyruvate will turn to acetaldehyde, which is 2-carbon compound. Okay, pyruvate will first turn to acetaldehyde and 1 carbon dioxide is eliminated here. And here what is the enzyme that is catalyzing the reaction from pyruvate to acetaldehyde? It is pyruvic acid decarboxylase. Pyruvic acid decarboxylase. Next, 2-carbon compound, that is acetaldehyde, is then turn to ethanol, here NADH, which is released at the 6th step of glycolysis, is utilized over here. Okay, and it will release out the energy equivalent, that is NAD plus is released out as the energy equivalent, which is utilized for the glycolysis process again. Okay, so the net energy released is 0 by fermentation. There is no amount of energy that is released by fermentation. Only 2 net ATP that is being released in glycolysis is considered as the energy released during fermentation. So if you consider the total amount of energy that is released during process of fermentation, it is less than 7% of energy. 7% of energy is released by fermentation process or by anaerobic respiration. Fine, right? Clear till here? Any doubt anywhere? So here what happens, ma? Pyruvate will first turn to acetaldehyde, and the enzyme that is catalyzing is pyruvic acid decarboxylase, after which acetaldehyde will be turning out to the product, that is ethanol, and this step is catalyzed by pyruvic acid dehydrogenase or alcoholic dehydrogenase. Alcoholic dehydrogenase is the enzyme. Fine, right? Where carbon dioxide is being released along with ethanol. What are the end products that are formed after alcoholic fermentation? It is ethanol and carbon dioxide. Fine, right? And is there any amount of energy that is released over here? No. NADH, whichever is released during glycolysis, is being utilized here, and this will give out the energy equivalent, that is NAD plus, which is utilized again for the glycolysis process. Total amount of energy release is just 2 ATP, that is also from glycolysis. So you can pass out one statement, which is the most prominent statement from your text as well, can be asked in statement based question, that is less than 7% of energy is released during anaerobic respiration. Clear? Next, lactic acid fermentation. Lactic acid fermentation. Here we go. First, glucose turning to pyruvic acid. Till here the story is clear to everyone. Pyruvic acid will be turned to lactic acid, and this step is catalyzed. Both are 3 carbon only, and this step is catalyzed by pyruvic lactic acid dehydrogenase. Lactic acid dehydrogenase. And the cofactor present over here for this enzyme will be zinc. Zinc is the cofactor. And here for alcoholic dehydrogenase and pyruvic acid dehydrogenase, the cofactor will be magnesium. Remember, the cofactors important, can be asked in connection with enzymes concept also. Okay, lactic acid fermentation, cofactor will be zinc, and here also NADH is utilized for the process, and the equivalent NAD plus is being utilized for glycolysis again. Clear till here, ma? Any doubts anywhere? And then, so this is all about anaerobic respiration. How aerobic respiration is channelized after pyruvate release. We have discussed, right? Immediately the pyruvic acid will move on to the mitochondrial matrix. Immediately from cytoplasm, pyruvic acid will enter into the mitochondrial matrix, and inside mitochondrial matrix, what is the first step that is happening? First step is oxidative decarboxylation. Okay, just a second. Yeah, it is oxidative decarboxylation is the second step that is being operated inside the mitochondria. Here in oxidative decarboxylation, 2 pyruvic acid molecules, let us count for 2, but a single cycle will have 1 pyruvic acid. Okay, for 1 glucose if you have to calculate, then calculate for 2 cycles of oxidative decarboxylation. Let us assume 1 pyruvic acid, or for 1 glucose if you see 1 glucose will give out 2 pyruvic acids. 2 pyruvic acids entering into the mitochondria will utilize, NAD plus, 2 NAD plus and 2 acetyl CoA or 2 coenzymes A. Sorry, acetyl CoA is formed as a product, right? So in the reactant along with NADH, 2 coenzyme A is being utilized, and this will react and form out to give 2 acetyl CoA, 2 acetyl CoA and 2 NADH plus H plus is being released out. And what is the enzyme that is catalyzing this process? Pyruvic acid decarboxylase will be the enzyme, or pyruvic acid dehydrogenase, any of the enzyme. Okay, pyruvic acid decarboxylase or pyruvic acid dehydrogenase. Fine, right? Along with 2 acetyl CoA, it will be giving out 2 carbon dioxides. 2 CO2 will be released out. Clear till here? Any doubt anywhere, ma? Oxidative decarboxylation step, I hope this is clear to everyone. Now we will move on to TCA cycle. Yeah, TCA cycle, here the immediate process that happens is 4 carbon compound, that is oxaloacetic acid, which is 4 carbon compound, will be accepting the 2 carbon compound, that is acetyl CoA. And remember, acetyl CoA will be the common intermediate for any of the respiratory substrates. If be it fats, be it proteins, be it carbohydrates, the common respiratory substrate will be acetyl CoA. In amphibolic pathway, we will learn it more clearly, but as of now, remember this statement. Okay, so this 2 carbon compound acetyl CoA along with 4 carbon compound, this will be forming out to be 6 carbon citrate is formed by the enzyme citrate synthase. And hydrolysis, dehydrolysis, both the reactions will happen, utilization of 2 water molecules and releasing out of 2 H2O. Here again I am telling you, we are considering for 1 glucose. 1 glucose. 1 glucose will give out 2 pyruvic acids. It will undergo 2 oxidative decarboxylations, 2 ODs, and then it will give out 2 acetyl CoA. Okay, this 2 acetyl CoA will be giving out 2 tricarboxylic acid cycles will happen. Fine, right? Remember, so in a single cycle only 1 acetyl CoA is involved. For 2 cycles 2 acetyl CoAs will be involved here. So here 6 carbon citrate will be turning to isocitrate, which is 6 carbon, by the enzyme aconitase. Okay, and isocitrate, which is 6 carbon, will undergo decarboxylation reaction. Okay, here decarboxylation happens, first decarboxylation, and it will release out carbon dioxide. Sequential release of carbon dioxide or sequential oxidation steps will happen here. Okay, so here carbon dioxide will be released and 6 carbon will turn to 5 carbon alpha ketoglutarate, and here if you consider for 1 glucose and 2 acetyl CoA, 2 NADH will be released. 2 NADH at the first decarboxylation step, followed by alpha ketoglutarate to succinyl coenzyme A, second decarboxylation reaction, where 5 carbon is turned to 4 carbon. Remember, decarboxylation steps from TCA are very important. Okay, if you look into the previous year questions also, the hotspot reactions of TCA cycle are this decarboxylation steps only. Questions are mostly picked from this area. Okay, so pay additional focus and listen. Alpha ketoglutarate, which is 5 carbon, is turned to 4 carbon, that is succinyl CoA, and here NAD plus is converted to NADH. Again NADH will be released into the cytoplasm, and the enzyme over here is alpha ketoglutarate dehydrogenase. 4 carbon acetyl CoA is turned to 4 carbon succinate. Here again substrate level phosphorylation will happen, where succinyl coenzyme A is turned to succinate. Here ATP is being released, ATP or GTP is being released out, and phosphate group is being taken from the substrate. Fine, right? Clear? Understood? And then succinate to fumarate by the enzyme succinate dehydrogenase, which is attached to inner mitochondrial membrane. Remember this point clearly, because this will be used for electron transport system. Okay, this is by the enzyme succinate dehydrogenase, where FAD plus is turned to FADH2. Fine. And then fumarate to malate, which is 4 carbon, again by the enzyme fumarase, and then malate to oxaloacetic acid by the enzyme malate dehydrogenase. Okay, this is the last step of TCA cycle, where NADH will be released. How much NADH? Again 2 NADH will be released. Clear till here? Any doubts anywhere? So here if you look into the bioenergetics of TCA cycle. Bioenergetics of TCA cycle, if you observe, first step, that is isocitrate to alpha ketoglutarate, NADH is released, alpha ketoglutarate to succinyl coenzyme A, one more NADH, and third spot malate to oxaloacetic acid, which is last step of Krebs cycle, again NADH is released. So at 3 points 3 NADH is released in one TCA cycle. If you consider for one glucose molecule, then 2 TCA cycles will happen. So for one glucose 3 NADH will be released. So 6 NADH. For one glucose, 6 NADH is released into the mitochondrial matrix. And how much amount of FADH2? From succinate to fumarate, it is 2 FADH2. And how much amount of ATP? That is from succinyl coenzyme to succinate, 4 carbon. How much amount of ATP? 2 ATP or 2 GTP is being released. 2 ATP or 2 GTP. This is from one glucose molecule. This is for one glucose molecule. Clear till here? Any doubt anywhere, ma? Okay, clear, right? So 6 NADH, 2 FADH2 and 2 ATP for one glucose molecule. Now, ATP is of course the cellular energy, we know. Cellular energy. But NADH and FADH2, they are the complex energy molecules which cannot be utilized for the metabolic processes of the cell. This has to be converted to the simplest form of energy, that is ATP. Fine. Now to convert this complex energy to the simplest form of energy, there is a last process that is operated in aerobic respiration, that is TCA cycle. Look at this diagram carefully in the TCA cycle. Yeah. Last concept of today's session. So here TCA cycle, listen it carefully. Sorry, electron transport system. Electron transport system. Where is this operated? Electron transport system is operated in inner mitochondrial membrane, that is cristae. Okay, this is observed in cristae or inner mitochondrial membrane. Fine, right? okay, observed in cristae or inner mitochondrial membrane, where 4 complexes will run up this process, okay, that is complex 1, which is NADH dehydrogenase, complex 2, that is succinate dehydrogenase, complex 3, which is cytochrome BC complex, complex 4, which is cytochrome C oxidase. Fine, right? So it is NADH dehydrogenase, succinate dehydrogenase, cytochrome BC complex and cytochrome C oxidase. Now where do you see the complex energy molecules inside the mitochondrial matrix? So all these complexes are channelized from mitochondrial matrix. They are channelized from mitochondrial matrix to intermembrane space, and the complete process is taken over by cristae, that is inner mitochondrial membrane. Look at the complex 1 carefully, ma. First, when NADH plus H plus is oxidized to NAD plus, what is that released? NAD plus plus H plus and electrons. 4 protons are directly pumped from mitochondrial matrix to intermembrane space. Protons will never go alone, they will move along with the electrons. So the electrons are being taken by flavin mononucleotide, that is FMN protein molecule from complex 1. Okay, so here from complex 1 you remember this complex 1, that is NADH dehydrogenase, is transpanning both the sides of inner mitochondrial membrane. This is transpanning both the sides. So here the NADH immediately after oxidation, protons are pumped from matrix to intermembrane space and electrons are being received by the protein molecules inside the complex. First it is FMN and then ferrous sulfur complex, and then electrons are being given to a mobile electron carrier. Okay, it is being given to a mobile electron carrier, that is ubiquinone. Okay, and then accepting electrons, it will be turned to ubiquinone, hydroubiquinone. Fine. Now complex 2, that is succinate dehydrogenase. Look at here carefully. Complex 2, which is succinate dehydrogenase. There only I told this enzyme is in close proximity or inside the inner mitochondrial membrane. But if you see complex 2, this is not transpanning both the sides of the membrane. This is just over to the side of mitochondrial matrix. So here after oxidation of FADH2, where FAD plus electrons and protons will be released, right, where protons and electrons both are being carried by the electron molecule, by the protein molecule, that is FAD. Flavin adenine dinucleotide will carry on or take over both protons and electrons, and then it will submit to ferrous sulfur complex and then to mobile electron carrier, that is ubiquinone. Clear? And here it will give out both electrons and protons to hydroubiquinone. From complex 1, electrons are being given. From complex 2, both protons and electrons are being given to ubiquinone. Fine, right? Now in complex 3, which is cytochrome BC complex, cytochrome B will be accepting electrons from complex 1, electrons and protons from complex 2. Clear? Is the story clear till here? Now the electrons and protons are being given to ferrous sulfur complex and then to mobile electron carrier again, cytochrome C, which is the mobile electron carrier, to which electrons are being given and protons from complex 3 are pumped to the intermembrane space again. Here cytochrome C will be carrying out the electrons. Cytochrome C is not in between two complexes. It is on the outer side of the inner mitochondrial membrane. Remember that. The positions of ubiquinone and cytochrome C are very important. Ubiquinone is present in between complex 1 and complex 3 and complex 2 and complex 3. Whereas cytochrome C is in between complex 3 and 4, and this is exactly located on the outer side of the inner mitochondrial membrane towards intermembrane space. And what is that which is immediately accepting the electrons in complex 4? It is cytochrome A and then copper molecules. Copper molecules and cytochrome A will be accepting, and from the complex 4, the ultimate electron acceptor is oxygen, which is located in the mitochondrial matrix region. Okay, here the ultimate or the last electron acceptor of ETS in mitochondria is oxygen. There, if you once remember the ETS, electron transport system, or non-cyclic photophosphorylation of photosynthesis, the ultimate electron acceptor will be NADP+, and here in the ETS, the ultimate electron acceptor will be oxygen. Okay, wherein it will be forming out what? Water molecules. Accepting the electrons, this forms out water molecules. Clear, right, in complex 4. Fine. And now this also transmits out the protons. Complex 4 also transmits out the protons. Immediately after the formation of water molecule, 2 protons will be released into the intermembrane space. So now if you see at the end of the chain reaction after complex 4, water molecule will be formed. Oxygen is accepting the ultimate electron, but now proton gradient inside the mitochondrial matrix, protons are decreased, and in intermembrane space proton gradient is high. High number of protons are present. Now in this case, what happens? Due to this proton gradient which is generated, F0 F1 complex will be activated, which is also known as complex 5. Complex 5 is also known as ATP synthase or F0 F1 complex, which is also known as Fernandez-Morgan particles or oxysomes. These are also the synonyms of this. So here ATP synthase, if you observe, in single run of ATP synthase, F0 F1 complex, F0 is embedded inside the inner mitochondrial membrane and F1 is placed towards the mitochondrial matrix. So in a single run, 4 protons are pumped from intermembrane space to mitochondrial matrix, and 4 protons are required for the formation of 1 ATP. For the formation of 1 ATP, 4 protons will be required. Clear till here? Any doubts anywhere, ma? Fine, right? So this is the complete process of ETS, and this is how the ATP energy is being formed. Clear till here? And then, yeah, here respiration is said as an amphibolic pathway. Okay, respiration is said as an amphibolic pathway. It is because the respiratory intermediates which are formed in the aerobic respiration are also the starting reactions or the starting steps in many biosynthetic reactions. Fine, right? Be it the biosynthetic reactions of hormones or be it the biosynthetic reactions of secondary metabolites, the respiratory intermediates here will start up the anabolic reaction, will start up the synthesis of the other process. That is the reason respiration is also addressed as amphibolic pathway. And as I told you in the beginning of TCA cycle, acetyl-CoA is the common respiratory substrate, is the common respiratory intermediate for any of these substrates. If you consider proteins will be deaminated to amino acids and they will be converted to pyruvic acid. From pyruvic acid it will join the aerobic respiration cycle. If you consider fats after fatty acid oxidation, it will be turned to fatty acids and glycerol. Glycerol will enter the glycolysis process as dihydroxyacetone phosphate DHAP by isomerization reactions, and fatty acids after oxidation process it will join the cycle through acetyl CoA intermediate. So be it proteins or fatty acids or glycerol, last the respiratory intermediate will be acetyl CoA only, and glucose there is no much explanation required now. Till now we are exercising this process itself. So here acetyl CoA, if you see after entering the Krebs cycle and then ETS will happen. After ETS ATP energy is being formed, cellular energy is being formed. Clear till here? Any doubts anywhere? Fine, right? Clear with amphibolic pathway? Now, once we will see total amount of energy required, total amount of energy released during aerobic respiration. Anaerobic respiration, it is 2 ATP, we have done it. Now, total amount of energy released during aerobic respiration. Fine. So here we see first during glycolysis process. How much amount of energy is released, ma'am? Yeah, it is 2 ATP and 2 NADH, right? And the second step, that is oxidative decarboxylation. It is 2 NADH. And then third, it is TCA cycle. Fine, or Krebs cycle. How much amount of energy that is released in TCA or Krebs cycle? It will be 6 NADH at 3 steps, right? 2 FADH2 and 2 ATP. Fine, and this is the complete amount of energy that is released. Now let us convert this to the cellular energy, that is ATP. Cellular energy. Here we go. It is 2 ATP and 2 into 3, right? 1 NADH, we will approximately calculate it as 6 ATP. Here is a query, just a minute. You continue watching, beta. You continue watching, get clarity on how much bioenergetics are released from aerobic respiration. You get this clarity. In 5 minutes, you hold on, you will get it clear. You will have a clear picture. From oxidative decarboxylation, it is again 6 ATP. Yeah. If you consider TCS cycle, 6 NADH, right? 6 into 3, 18 ATP and 2 FADH2. Here 1 FADH2 we will approximately calculate it as 2 ATP, right? So 2 into 2, it will be 4 ATP. Then again at last 2 ATP. Here we go. So on total, if you calculate, ATP will be in total. Yes or no? 38 ATP in total. For aerobic respiration, it is on total 38 ATP. But here is a thing that you have to note, that in a eukaryotic cell, the amount of energy that is being released from NADH, one NADH to get transported into the, here once you have a look at here, yeah, here in glycolysis, when 2 NADH and 2 ATP will be released out, right? This NADH, to get it translocated to mitochondrial matrix, inside inner mitochondrial membrane, shuttle systems will be operated. Okay, glyceraldehyde shuttle systems will be operated. So those shuttle systems will take over 1 ATP. 1 ATP is charged to transport 1 NADH from cytoplasm to mitochondrial matrix. As such, for 2 NADH, how much ATP will be charged? 2 ATP will be charged. So when the shuttle system is charging 2 ATP, from the total energy that is being released, that is 38 ATP, just eliminate out minus 2 ATP, which are utilized by what? Shuttle systems to transport the NADH from the from cytoplasm to mitochondrial matrix. So in a eukaryotic cell, the total amount of energy that is released at the end of aerobic respiration will be 36 ATP. Total amount of energy released in a eukaryotic cell is 36 ATP. Fine? Clear till here? Any doubt anywhere, ma? You are clear on the complete bioenergetics of aerobic respiration? Clear on amphibolic pathway? Fine? Now, we will see last concept of the respiration in plants, that is respiratory quotient. What is respiratory quotient? The respiratory quotient will tell you the volume of carbon dioxide evolved is to volume of oxygen consumed. Respiratory quotient will give you the ratio between carbon dioxide evolved and oxygen consumed in the aerobic respiration for different respiratory substrates. If fats are the respiratory substrates, here in our text one example is given, that is tripalmitin. Here the amount of carbon dioxide released is 102 and oxygen consumed is 145. More amount of oxygen is required to oxidize the fat molecules, right? So here it will be less than 1. Respiratory quotient will be less than 1 because denominator will be more than the numerator. Here for fats, let us draw out a table for respiratory substrates is to RQ value. Fine. First for fats, it will be less than 1, that is 0.7, and this is the reason. And if you take carbohydrates, the amount of carbon dioxide evolved and will be equal to the amount of oxygen consumed. So here for carbohydrates, it is 1. If you consider proteins also require more amount of oxygen for oxidation. So third, if you take proteins, RQ value is 0.9. And then in anaerobic respiration, if you consider anaerobic respiration where no amount of oxygen is utilized to oxidize the carbon dioxide. So something by infinity, so something by 0 will give you infinity. For anaerobic respiration, it is infinity. Fine, right now. So this ends the complete chapter. Once let us have a quick recap on what we have learned. starting from respiration and respiratory substrate. Cellular respiration is clear to us. We have looked into types of respiration based on respiratory substrate and presence of or absence of oxygen, followed by glycolysis, anaerobic respiration, followed by oxidative decarboxylation and then Krebs cycle. Lastly, ETS, amphibolic pathway and respiratory quotient. Fine, right? So the complete chapter is summed up in a single hour. This you can watch it as one shot as well. Fine. And then the next chapter that we are going to exercise is plant growth and development, where phytohormones are the hub of that chapter. So see you in the next class again.